Is someone absent today, is it? On Nurin. Okay, can someone keep on Nurin? Okay, keep on Nurin now. Okay, make sure you write her name down on the paper. Can I have the extra copy? Okay, can pass the extra copy to Aida or Nanya? Okay, now I'll give you some time. Please try out the crossword puzzle. See how much you can recall. Okay, you can work with your friends. Okay, now play some nice music. Okay, now I'm going to play a now it's a hotline. Now hotline. Okay, I want to refer you once again. Lumos is a rich resource. Okay, I've already found videos that will help you out. Okay, so I'm going to play a video. For those that still have trouble, this video may help you out.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called a nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, I will not review the answers yet. I'll let everyone take a pause. We're going to watch a video to link everything together. And I think you will start to fill in the blanks in your mind, the ones that you couldn't quite find in the textbook. Okay? This chapter is best learned by watching videos. Reading the process for the first time is very messy in your mind, in my mind at least. But when I watch a video, actually, it's a very clear process. Okay, I'm going to show you a video now. Okay, to try and make sense of transcription and translation. Ready? Yeah, so if you are still doing, I'd like you to pause for a while. Okay, just give this a watch, right? To link all the process together. The blanks will be filled in. Ready? Yeah, everyone heads up. Okay, you will fill in the blanks in a while. Okay, let's watch out. Okay. Find your answer through this video.
Proteins are made of a long chain of amino acids, which has been coded for by DNA. The order of the bases of DNA determines which protein is made by coding for a specific order of amino acids. This is protein synthesis, and in this video, we're going to look at how it works. You may want to watch our What is DNA video first to remind yourself all about DNA. A set of 3 bases in the DNA molecule codes for a particular amino acid. GGT, for example, codes for the amino acid glycine. The 3 base codes are known as codons, so GGT is a codon for glycine. So there are 20 amino acids that make up our body's proteins. 9 of these we need to take in through our diet, as our body cannot manufacture them. These are known as essential amino acids. Our body can, however, manufacture non-essential amino acids. Whilst 20 amino acids build up proteins, there are actually many others that do not form proteins, possibly over 250. They may form sugar, for example. Before we start, you need to know what RNA is. So you may want to watch our What is RNA video first. So let's now look at how proteins
So let's look at
actually
transcription and translation
synthesized.
now.
The DNA is in the nucleus and cannot move, but the ribosomes in the cytoplasm are where the proteins are made. This means the code from the DNA needs to be copied and carried across to the ribosome by a molecule called messenger RNA, or mRNA, before the protein can be made. Let's have a look at how all this works. In the nucleus, the enzyme RNA polymerase unwinds and unzips the 2 strands of DNA that contain the protein-making gene. Only one of these strands is going to be replicated. Complementary RNA nucleotides base pair with the chosen strand. RNA polymerase also then binds the RNA nucleotides together, making a new RNA strand. This is called messenger RNA, or mRNA, and this whole process inside the nucleus is called transcription. The mRNA then travels from the cell nucleus and out into the cytoplasm until it reaches and attaches to a ribosome. The ribosome then sticks the amino acids together to make a polypeptide chain, following the order of amino acids as coded for by the mRNA. 3 base codons on the mRNA code for 1 amino acid, and this whole process in the ribosome is called translation. So let's look at translation in a little more detail. What is actually happening inside the ribosome? Transfer RNA, or tRNA molecules, carrying specific amino acids base pair with the mRNA inside the ribosome. So the tRNA brings in the amino acids, and the mRNA provides the information of the exact order that the amino acids need to be bonded together to ensure that the correct protein is made. As more and more tRNAs match up with the mRNA, the amino acid chain becomes longer and longer, and eventually the polypeptide chain will form into the protein. So there we have protein synthesis. Inside the nucleus, the DNA molecule is unzipped from around the gene by DNA polymerase. The DNA is then replicated and mRNA is formed in the process of transcription. The mRNA has to travel out of the nucleus and into the cytoplasm, where it binds with a ribosome. In the ribosome, the nucleotide sequence of the mRNA is translated by the tRNA, which carry the related amino acids.
Okay, so who's left with some blanks that you cannot find? What number is that? What number is that? You raised your hand, right? Oh, I had a question. Oh, okay, what's your question?
What happens to the mRNA after the translation is done?
Okay, Arish asked a question. What happens to mRNA after translation is done, is it? Sorry? Okay, so what happens to mRNA after translation is done? Yeah, the messenger RNA. Okay, everyone think, does the messenger RNA become a protein? It's just a recipe page. Right? It's just a recipe that the ribosome reads to create a protein. So what do I do with that recipe? So many things can be done. You can either break it down or you could reuse it. Another ribosome can come and create a new protein from it. You know the vaccines they are taking? One of the things they are injecting into you is mRNA. So they are injecting into you mRNA that when your cells read will create parts of a virus inside you. Right? So you are actually creating parts of the virus when you are being injected with the mRNA. But your cell's immune system will see the parts of the virus and they will start to create an immune system response, right? Without having to face a real virus at all. Okay, so to answer your question, mRNA is reused or is destroyed. Okay, so let's run through the options. I'm going to go through in a different sequence, but so it's in the sequence of transcription and translation. So, first and foremost, where does transcription take place? It takes place in the nucleus. You want to go wash up? Okay. So those a bit sleepy, you want to go wash up, Transcription. Okay, where does transcription take place? Question 7. It takes place in the transcription. It takes place in the nucleus. Okay, next. 8. Where does translation take place? Cytoplasm. So you need to know where these two processes take place. Okay, they take place in different regions. What's interesting is that bacteria has no nucleus, right? So transcription, translation takes place all in the cytoplasm. Okay? Alright, next. Transcription, number 3. Transcription begins when RNA polymerase recognizes the question 3. Transcription begins when RNA polymerase recognizes the promoter. Yeah, you're right, promoter. What you're right? Missed out an alphabet. No, it's not a prompter, it's a promoter. Okay, so it's a promoter. Yeah, it's a promoter, not prompter. You all spell wrongly. Okay, so, next question. Question 2. The template strand is transcribed to form mRNA by RNA polymerase. That's the enzyme that creates the mRNA after reading the template strand of the DNA. Okay, next, question 6. mRNA is synthesized using adenine, cytosine, guanine, and uracil. So RNA does not use thymine. Next, question 11. Transcription ends when RNA polymerase encounters the question 11. Yeah, I even see the question number, okay? Question 11. Transcription ends when RNA polymerase encounters the Terminator, the movie. Okay? Okay, so there's one thing I'd like you to take note of. Number 1, if you talk about transcription, it starts when it recognizes the promoter, and it stops when it recognizes the terminator. And this takes place in the nucleus using the enzyme RNA polymerase. Now you have created an mRNA. Okay? So let's move on to the next place. Where does translation take place? Okay, so it takes place in the cytoplasm. That's question 8. Okay, so question 14. Translation requires what to transport amino acids to the ribosome? tRNA. Okay. That's question 40. Question 1. tRNAs are able to complementary base pair to its corresponding codon using its anticodon. Thank you. Using its anticodon. Question 12. Translation is carried out by ribosomes. Okay, so here what we have RNA polymerase. Ribosomes is the one that carries out translation. Next, question 13. The first codon, the mRNA to be translated, is called the start codon. Okay, so for translation to take place, we need to find the start codon. Okay, not the promoter. Okay, so usually students will confuse between the two. They will think for translation to take place, the ribosome needs to find a promoter. That's for transcription. Okay, so there are different processes. Okay, so let's end translation. Question 10. Translation ends when the ribosome encounters the stop codon. Okay, so now, right, so far what we've learned is this. For both processes to take place, there are some key players that are involved. Here's an enzyme, here's ribosome. For both processes to take place, you need to somehow recognize the start and recognize the end. You need to recognize the start and recognize the end. Right? Okay. Anything else? Number 4. Genetic code is read in triplets of bases known as codons. Am I left with anything else? Question 9. Ribosomes function to form what bonds between peptides? Peptide bonds. Okay, I think we covered all of it. Okay. There is the first amino acid, is it? Okay, what's the first amino acid to be translated from the start codon is called? How to pronounce that? Methionine. No, it's methionine. Methionine, not methionine. Okay. I would like to run through the game that you played one more time so that we can see all of this in action. I always believe transcription translation, if you read the process, it's very hard to visualize. But if you watch the process, it's much more easy to understand. Okay, so let's use all these terms, but now in the simulation. So you've done this, right? Okay, who here just watched the first 40 seconds of video, is it? Yeah. Some students only thought that there was a video. Okay, so I'm going to play the video. Okay, click to start. Okay, click to start. Now let's play together.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
Right, and then this is the point where the real game begins. Okay, so you can now pick a gene that you like to transcribe. Right, I hope you did that. Okay, so I'm going to pick a gene I want to transcribe. Okay, so on the whiteboard now I have DNA. As we all know, DNA is double-stranded. Do you remember DNA is also anti-parallel? Okay, so this strand over here, the top one, does it run from 5 to 3 or 3 to 5? From left to right? Left to right, top one, 5 to 3 or 3 to 5? Okay, how do you identify the 5' end? Okay, if you see a P, that is the 5' end. Okay, if you see a P, this is the 5' end. If you see a OH, that is the 3' end. So this is the 3' end, this is the 5' end. This is why DNA is anti-parallel, right? Do you remember DNA replication? When you want to create DNA, do you add new DNA to the 3' end or 5' end? 3.
The hydroxide.
Okay, cannot remember all this, huh? Okay, so I'm going to take a pause here right now. Let's say I want to create an mRNA. I need you to follow me here. Okay, so the first thing, right, is RNA polymerase as an enzyme comes in, right? Okay. What does RNA polymerase need to find? It needs to find something called promoter. Okay, the promoter here. So let's say RNA polymerase comes in like, oh, okay, I found the starting point. Okay, I'm going to anchor myself here. I'm going to get ready to do a copy. What RNA polymerase does next is quite interesting. You will then unwind the DNA. Okay, you'll unwind the DNA. Yes. Okay, so Bansi remembers helicase also unwinds DNA. Right? But we find that DNA replication, helicase can only do one job, which is to unwind. RNA polymerase can do two things. It can unwind and it can copy. Okay, it can make mRNA. So RNA polymerase can do two things. Okay, so now I will need your help. I'm going to create mRNA now. Okay, let's create mRNA. One of the strands will be called the template strand. Another strand will be called the non-template strand. So I'm going to label for you now. This strand on the bottom, I call the template strand. The one on the top, this is a non-template. The strand that the RNA polymerase uses to create mRNA is called the template strand. Okay, there's a meaning to the words now. Okay, so imagine the enzyme now comes and it starts. I'm the enzyme now. I'm RNA polymerase. Okay, found you, unwind. Now I'm going to create mRNA. Okay, now I need your help. If I want to create mRNA, complementary base pairwise, what should I put on top? Okay, A. Okay, let's put an A. Done? Right, we've done one of them. This is the 5' end, this is the 3' end, right? Okay. Now let's elongate the mRNA. What's the next one? U. Okay, let me find U. Okay, let me find U. Wait, where's my U? I have lost U. Okay, I found U. Okay. Okay, now notice this, when the new mRNA polymerase, I will add the new RNA to the 3' end. Yeah? So this is the new 3' end. The next one, this one will be G. Okay, let's put a G. Okay, next, this one will be G. Okay, let's continue on. A G. Can you describe for me the direction in which I'm building the mRNA? I'm building in the 3 to 5 direction. Okay, can you see I'm building the mRNA in the 5 to 3 direction? I've been adding it to the 3 end, the 3 end, the 3 end. Ready? I'm building it, I'm adding it to the 3, I'm adding it to the 3 end, I'm adding it in the 3 end. I'm building it in a 5 to 3 direction. Do you notice in your textbook they mentioned this? They build the mRNA in a 5 to 3 direction. I'd like you to refer to page 52, 53. If you can find that term, I'd like you to highlight it. mRNA is synthesized in a 5 to 3 direction. Which page is it on? 52? 53 also have okay, 53 is the diagram, 52 is the words. Okay, I'd like you to highlight it up. It's important to know the direction in which mRNA is built. Okay, it's built in a 5 to 3 direction. How would I describe the way in which I am reading the template strand? Can I say again? Okay, run, follow me up. If I am building the mRNA in the 5 to 3 direction, I am reading the template in the what direction? 3 to 5 direction. Okay? So I read the template in the 3 to 5 direction. Okay? I build I build the mRNA in the 5 to 3 direction. Okay? Okay. I would like you now to do, I often hear a lot of people say this, but I would like you to think about it. So, okay, let me transcribe the entire thing first. This is a G followed by a, okay, so C, let's build some more. A, U, okay, A, U, G, G, C, C. Okay, what? C, C. Okay, done. Very good. I created mRNA, right? Okay. Okay, after I created the mRNA, what RNA polymerase will do is, when it encounters, no, go up. Okay. When the mRNA encounters on the end of the gene, something called a terminator, it will then know that, okay, this is where I should stop doing the copying process. After it knows that it's reached the end, what will it do? We'll just close up the DNA. Let's close it back up. Okay. And then I will bring the mRNA. I'll bring the mRNA to the cytoplasm. Okay, let's bring it to the cytoplasm now. Where ribosome will come in. Okay, so now I have mRNA. This is the template. This is the non-template. Okay, I'd like to introduce to you one new term. Sometimes people call the non-template strand the coding strand. But I want you to try and make sense of the word. Why do you think it's called the coding strand? Okay, what's the same? Sequence of? Which nucleotide? Okay, let's be specific. I hear that something is the same as the coding strand. What's the same as the coding strand? Everything of what? The template strand, the mRNA? No, what's the same? Okay, now I'm getting it. The coding strand and the mRNA, the sequence is the same. Right? You see, A, T, oh no, except the fact that it's a U, right? But you notice the sequence is the same. The mRNA sequence is the meaningful sequence, right? It is the sequence where the tRNA will try to recognize to bring amino acids. This sequence is not meaningful. Okay? It's just a complementary base pair to it. I feel I lost some of you. Okay, still following? Okay. Now I'd like you to, this is a brain teaser. A lot of people say this. I'd like you to see the extent of this. Okay. People always describe transcription. Transcription is a photocopy of the DNA. To what extent is this true? Okay, 5 marks. Okay, go on. Who's the history student? You history student? Okay. So to what extent is this true? Transcription is a photocopy process when we photocopy the DNA, the entire DNA. To what extent is this true? Okay, who says it's false? Who says it's true? Okay, so let's talk about it. Okay, so how true is this? Okay, let's pick out all the false parts of this statement. Transcription is a photocopy of the DNA. Okay. Okay, so what are we photocopying when we create mRNA? Actually, we were photocopying the coding strand in a sense, right? But we made reference to the template strand. So it's a bit weird. And then, it's not even a photocopy. I don't even take the T, right? I replace it with U. Okay, so it's not really a photocopy, right? Next, you look at mRNA. It's so much shorter than the actual length of the entire DNA strand. Right? It's like carrying a page out of a textbook. That's not a photocopy. So in many ways, this statement is not very true. Is there a process that is a photocopying process of DNA? Okay, so DNA replication is really a photocopying process of the DNA. Which phase does this take place in the cell cycle? Interphase, and more specifically, the S phase, right? S phase is when DNA replication occurs. That is the real photocopying process. Creating mRNA is not a photocopy process. Okay? Okay. So, understand that there's a template strand, there's a non-template strand, sometimes we call a coding strand, and we get mRNA. mRNA is synthesized in a 5 to 3 direction, and we read the template strand in the 3 to 5 direction. Okay? This is the important part that I would like you to focus on. Do I have time up? Okay. So let's play this game now. Now that we've cleared the tough part. Then you can visualize the tRNA part. The tRNA part is very cute. Okay, I need your help now to play this game.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases. RNA polymerase attaches to a gene.
Okay, they just say RNA polymerase attaches to a gene. Where does it attach to specifically? It attaches to the promoter. Okay, so it attaches to the promoter, then it starts everything. Okay, but it's not mentioned here. Not start codon, the promoter.
It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
Okay, let's transcribe. I need your help. Let's do this fast. Okay. G. Next. Come on, faster. Next. What?
When the end of the gene is reached, transcription is complete.
How does RNA polymerase know it's reached the end of the gene? Okay, the Terminator, the movie. Okay.
The new strand of mRNA passes out of the nucleus and into the cytoplasm.
Okay, so Ida is asking, what's the promoter? Right? Okay. I didn't elaborate, but the promoter is just a double-stranded sequence of DNA. Okay? It is not a protein, it's not a carbohydrate. If I want to be very specific, the promoter has a code usually of like something like that. Okay? So usually if I search a DNA, when I do research, right, if I see this sequence, I know that this is where RNA polymerase will bind to. Okay? Yeah, so after the terminator, there may be some empty space of useless stuff, and then after that, another promoter maybe. Actually, our DNA has a lot of garbage. We don't really know its purpose. We only know the purpose of some of them.
Translation takes place.
We don't know.
The instructions in an mRNA strand are made up of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning leaf protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid to build blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid thymine docks with the ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA, signaling the beginning of protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. Final shape of pride.
Okay. For you, I would like you to just double check one thing. Can you flip to your textbook where you find the genetic code? What page 55, right? Okay. Do you need to memorize the genetic code? Yes, I need you to memorize it. No, you don't. Of course not. I want you to apply, don't memorize. Okay? Okay, so I give you an example right now. If I give you the mRNA CGA, can you read this and tell me what amino acid is supposed to be brought in? You read the genetic code. Come. What was the amino acid? Arginine? Okay, do you see arginine? Okay, let's look at the tRNAs here. Which tRNA is carrying arginine? At the top right? Okay. How does this tRNA know that it needs to bind to this codon here? With the help of this anticodon here. Okay, see the anticodon is complementary to the codon. So now let's bring it in. Yay, one done. I would like you now to read the next codon, CAG. Can you find it? CAG on the genetic code? Blood. What's it called? Okay, GLN is it? Okay, let's find GLN. GLN. Okay, this one. So I need to bring this in. Alright, how does it bring it in? Okay, the fact that it has an anticodon that is complementary to this over here. If there's a match, then they can bring it in. Okay, so this is how we read the genetic code. The genetic code helps you read the mRNA, not the anticodon. Yeah, say again. The genetic code helps you read the mRNA, not the genetic codon. No, not the anticodon. Okay? Okay, so I will stop here because it's almost the end of the lesson. Which means I have to wish you all the best. For those of you who are going to take the oral exam today. Any words of advice for your friends going for the oral? Okay, if not, thank you class. I will see you next week. Thank you. Thank you. Thank you.